Okay, so a couple of things. I haven't spied on you guys yet, but couple of your classmates were commenting about the assignment, which right now is eluding me as to where it is. Where's the agreement for you guys? So most of you have just been getting it from the front page on your list of things to do, right? The agreement or grades, because I didn't put it in any of the modules. Okay, so remember this has to be done with Lockdown Browser. Get it done today. And so if you didn't download the Lockdown Browser at home, right, remember up here in Building 2 on the second floor, you can go to the computer lab and you can do it there. It's kind of a survey, but it is graded. So if you answer some of the questions based on what I clicked as the correct answer for the answer key, you're not going to lose points, because one of them is also a short answer question where you got to tell me something. So I have to go back and hand grade all these anyways. You'll notice you don't have a grade in the grade book even if you completed it, right? So I will do that this weekend. So get it done before I grade it, preferably today, right? Because after I grade it, I'm not going back and regrading it. Not happening. And I don't know if you guys noticed, because I don't remember if I told you, but I did grade you all's introductions, right? That was kind of cool. Some of you guys might have had comments for me. No one has replied, so I thought I might need to let you guys know about that. So I'm going to go to my student view. And this is true for both my online and my face-to-face class, that I graded the introduction. I've had it graded for a while now, actually. Oh, where's my student view? There it is. So when you go to your grades, okay, so as a student, this is a really important feature for you guys. So notice introduction, actually it even shows up on recent feedback. So I just added this comment, right? You didn't complete this assignment. So if you click on it, right, it'll go to what you submitted, which of course is nothing, because as a student I didn't do anything, right? And you'll see my comment, and then you could reply right here. The other way you get to it is grades, and notice since I just posted something new, it even gives you a little indication. So that's the little box I'm talking about. So some of you guys have this little box. I may have asked you a question, or I may have given you a clue as to why you got it wrong. And for this particular assignment, there is a rubric, which should also show up here. I don't know why it isn't, but when you click on it, the actual assignment, it shows up off to the side. So this is how I grade, right? This is basically the instructions, right? The instructions tell you get 4 points for putting your result, 2 points for explaining your result, 2 points for replying to a classmate, 2 points for replying to another classmate, for a total of 10 points. So if you didn't get your full 10 points, then you look at your rubric and you can see where you lost points, right? Or I made a comment, or I did both. Which I think in this class, I don't think anyone lost points. But if by some chance, right, in the future, if you lose points on a discussion board, you can figure out why. Either I put it in the comments, or usually if you lost points, I definitely put it in the rubric. Sometimes I also comment. Just depends on how I'm feeling at the time. Whether I feel like doing it twice, basically, in 2 different forms. So, you know, at any time you can comment on an assignment, right? So if I might comment like, you know, reply to one more classmate, email me, and I'll change your grade, right? So sometimes I'm nice like that, so you definitely want to look at these things so you can get your points. Okay, I usually definitely do that for the first discussion board. I usually give people an extra try. After that, if you can't follow instructions, I don't know what to do. So under modules 2, and this applies for both online and face-to-face, I did finally, early this morning, because I kept falling asleep last night, I'm sorry, I added the objectives for genetics, and then the changes that I've made to the first genetics PowerPoint. The first genetics PowerPoint goes with Chapter 7 in our current book, right? The next genetics PowerPoint, Part B, that I'll post, goes with Chapter 9, right? So if you want to get ahead in your reading and such, check out Chapter 9. This was the original that I had posted for you guys, but I would go by the B one, version B. It has all the necessary changes. So this is the objectives list for genetics. And so unfortunately, they don't talk about the chemical mutagen ethinium bromide in your current book. And so I put a little asterisk there, right? And says not covered in new book but referred to PowerPoint. So that there's one new slide for that in the PowerPoint that I posted and that I'll use. They don't talk about excision repair in the new book, so I've deleted that from our list of objectives. Direct repair they refer to as the photoreactive pathway, so I put that in parentheses. They also don't talk about SOS response that I could find anywhere in the book, so I've eliminated that. So we have started to look, we haven't looked at these mutations yet, repair yet. We started to talk about it. And then the, okay, this is in the next lecture. I was like, I forgot to put this in this one. Okay, so this will be in the new PowerPoint. They also don't talk about the Ames test. So that information will just be in your PowerPoint, not in your book, unfortunately. So just two things, not too bad. All the rest is covered in your book between the two chapters that we're going to refer to, Chapter 7 and 9, for all of our genetics. I'm just looking at this. This one is probably covered in your book, but it's kind of a, I do kind of a summary slide for that. It'll help you with the understanding of some of the information in Chapter 9. Chapter 9 is going to be a foreign beast to you guys. So my lectures are going to be really important. Go ahead and try and read. Do the best to try and understand this, but I'm going to hopefully try and make it understandable for you. Like hopefully I've done so far. So we left off at last time. I know we talked about frame shift, right? I did the big dog ate the cat example. We talked about this, right? So we were just about to identify the enzymes, which we've already talked about, that proofread. Correct, Lance? I'm good? Okay. Yeah. So the type of enzymes that have the ability to proofread DNA as it's being replicated. So this almost seems like a trick question, y'all, or a quick trick statement. What makes DNA? What enzyme? What class of enzymes? DNA polymerase. There's lots of different ones, DNA polymerase 1 and 2 or 3, depending on what organism, right, and what domain you're talking about. But all in general, they're DNA polymerase. They make DNA. Well, guess what? They don't just make it, they also what? They proofread it. They proofread their own work, and they can fix their own mistakes. Are they 100% perfect, never make a mistake? No, because we'd never see mutations. If they were perfect, right? But they're pretty darn close to perfect, right? So for instance, I think it's E. coli's DNA polymerase makes a mistake about 1 in every billion bases. Wouldn't that be awesome if you only made a mistake 1 in a billion times you do something? That would be really awesome. So the very low rate of mutations that we see, this spontaneous mutation, is due to the fact that DNA polymerase proofreads its own work. So, actually, I think we did talk about that, huh? Okay, and we talked about these chemicals changing and then base pairing, and you ending up with base substitutions because of that. Okay, we got further than I thought. That's always good to review. All right, so this is the brand new slide. Yeah. So this chemical mutagen is a specific one that a lot of scientists that do molecular biology would work with. So I have worked with this chemical known as athenium bromide. It's in a class of chemical mutagens known as intercalating agents. And what that means is, as you can see in this picture, is that it binds to the DNA in between the bases. So remember when we talked about the structure of DNA and how important it is? Part of the reason why it's so important is because that enzyme, DNA polymerase, literally hooks on to the sugar phosphate backbone and reads the nitrogenous bases from that template strand to produce the other strand, right? And so does RNA polymerase. It attaches to the sugar phosphate backbone and it slides down it and reads that code. Well, when it gets here, remember how we talked about the width of the steps needing to be perfect in that beautiful spiral staircase that we envision DNA as being? That sugar phosphate backbone, right, that creates these lines would be like the railing to that spiral staircase. Well, imagine if you're like a little kid sliding down that banister. That's pretty much what this enzyme is, what these enzymes do. Now, if it gets to a point where, say, the carpenter missed a step, right? So there's this big gap here. He didn't put in one of the steps. DNA polymerase knows that after a certain distance there should be a nucleotide there, right? But there isn't, because this intercalating agent is inserted itself in between, and there's this extra space here. So what is DNA polymerase going to do? Well, just like you would tell your carpenter. Yeah, fix it. Put another step in there, right? I don't want to have to step up that high. Thank you very much. I want nice, even, steps all the way up my beautiful spiral staircase. So, it's like, okay, there's supposed to be something there. So I'm going to put something there. Is that good? Not really, because we know if we basically, in this case, insert a base, what's going to happen to the code in that region? If you add a base, what's going to happen to the code in that region? It's going to get shifted, right? That's going to cause what type of mutation that we talked about? A frame shift, right? Adding or deleting bases will shift the reading frame. So it's going to add it, but the bad news here is that now it's going to shift our reading frame, right? Everything from that point on is probably going to be messed up within that gene. What effect would that have on the cell? Depends on how important that gene is, right? Could be good. Yeah, probably not. Right? With this type of mutation, most of the time, frame shifts are not good. Could be lethal. Cell could die. So I was trying to find it on the internet this morning. I was trying to find a picture of the bottle of athenium bromide. When I worked in the lab, although I think they've changed the labeling, the material safety data sheet, and all that codes, but it literally had a skull and crossbones on the bottle, right? Because it is a known mutagen. We know it does this to the DNA. We know it mutates DNA. So why was I using it in the lab? I wasn't causing mutations with this chemical mutagen. I was using it for another purpose. For the reason that it binds specifically to DNA and RNA. So the unique thing is that when it binds and you shine UV light on it, it fluoresces. So when we take pieces of DNA and we cut them up with things like restriction enzymes and we put them in a gel matrix that separates them out by size using the electrical current, then you get these bands on a gel. Well, that's, maybe thousands, maybe millions of copies of DNA. But you still can't see DNA with your eyes, can you? We need something to help visualize it. And that's what actinium bromide helps do, is that it binds to the DNA, and when you shine the UV light on it, it fluoresces. So this band here, these lines represent where the DNA is in the gel. And its location in the gel is based on the size of that piece. There are other things that we use nowadays to dye DNA that's not supposed to be as toxic or dangerous as actinium bromide. And in fact, here at Delgado, we're not allowed to use actinium bromide because of this, right? Because of the fact that it will mutate your DNA. There was a colleague of mine in my lab, he never wore gloves when he worked with this reagent. It's like, dude, you're crazy. I am not mutating my skin cells. He's like, most of the ones on the outside of your hand are dead anyways. Your skin cells. I was like, yeah, I'm good. I want to have kids someday. He doesn't have kids. Maybe that's why. He was also a chain smoker.
So you don't use it in lab or here at Delgado?
No, other labs do still use it.
But
we don't here because of the potential of how dangerous of a chemical it is.
Right. So the question is, because you can inhale it?
No, it's liquid, usually in a liquid form. But, you'd have to wear gloves, right, because you don't want to get it on you, because it would do that to your DNA. It would bind to your DNA, and when you go to copy your DNA, right, it would be messed up. So this is another mutagen, an induced mutation. So these aren't ones that happen spontaneously, they're caused by something, right? That's what induced means. This is one that we're exposed to all the time, and this is why you want to try and protect yourself, UV light. It does this to your DNA. When you have two thymines next to each other, it can cause those two thymines to covalently bond with each other. What does that mean? Covalently bond. Chemistry people in here? What are they sharing when they covalently bond? Electrons. That's a really intimate exchange there, right? The electrons are being shared. Unlike this bonding that's supposed to happen across the strand where that's hydrogen bonding. That's a little bit weaker bond. The problem with this is, what else do you notice in this picture happens to the molecular structure of the DNA when the two thymines dimerize and bind with each other? Yeah, the sugar phosphate backbone's kind of bulged, huh? So remember what I said about those enzymes, how they slide down? So this is like when they put those bumps on escalators to keep kids from sliding down, the space in between the escalators, you know what I'm talking about? Those are actually deterrents for people sliding down the space in between the escalators. That's not architecture or anything. Okay? So, some banisters, right? Imagine if they put bumps on it. Obviously, you're not going to slide down it, or you're going to fall off when you get to the bump. Do we want speed bumps on our DNA? No, because the enzymes are going to fall off and not read that information, or they're going to misread that information. Do we want them to misread it and say, oh, well, I think there's just one T there? That'd be like deleting a base, right? Then you're going to end up with what? Frame shift. Is that good? No. So you're all going, oh my God, right? This happens to my DNA when you go outside and you're exposed to UV light. How come we're not all dead? Nope, it's always bad. Nope. Is this a permanent change in our DNA? No, it can be fixed. We have repair mechanisms for this. But you can only repair so much, right? You can only beat up on something so much. This is why over time, right, eventually the damage can't be repaired, and you end up with things like skin cancer. This is why the people who own those tanning bed places probably hate me to the core. Because I always tell my students, don't ever, ever go there. So what are those tanning beds? UV light. You're purposely exposing yourself to a mutagen. In high concentrations, no doubt. Not a good idea. If you must look tan, spray it on, or just realize like the rest of us have, white is absolutely gorgeous. Right? Because how many of you guys know Dean Gruber? The dean of our math and science division. Old guy in the office. Okay. If you walk by there, his nose is not so beautiful anymore. Right? He's had 2, maybe 3 surgeries on his nose to remove skin cancer. Because he was a baseball coach, and he spent hours upon hours outside in the sun without protection. And of course, he's an older gentleman, so it caught up with him eventually. Yeah. All right, so the good news, right, is that we can fix some of these things, right? But this is also why things like we use UV light to kill bacteria, right? High doses of UV light, we damage their DNA so much so that they can't repair it all, and we kill them. So at the molecular level, right, you need to know about these repair mechanisms. So what is direct? What is photoreactive pathway, and what type of mutations does it fix? And what's mismatch? How does that work? And what type of mutation does that fix? So we're only going to talk about 2, right? I've narrowed this list that was once 4 down to 2. So in your book, they go over mismatch repair. They talk about photoligase, which is the enzyme responsible for photoreactive pathway, also sometimes referred to as direct repair. And then we've already talked about DNA polymerase, ability to proofread its own work. Exonuclease, meaning that not only does it make the DNA, but it also can remove it and replace the nucleotides with the correct ones. So this next diagram was really small, right? And there was 2 of them next to each other, so I chopped them up and made them bigger so we could actually see. So this is something that we mentioned earlier, but let's see if you guys remember. So we know, because they're telling us, that this is the original strand of DNA that was used as the template, right? But what else other than that they're telling us and that this is, you know, highlighted in blue, how do we know chemically that this is the template strand, the original strand of DNA, and not the one that was replicated? What's on that strand? So my chemistry people, what is this? It's carbon and 3 hydrogens. It's a group known as a methyl group, right? So remember, we methylate our DNA when it matures so that we know, right, that that's the DNA that's been checked. That's what's going to be the next parent strand. So the 2 methylated mature strands of our parent DNA within the cell split to become the templates, right? And DNA polymerase makes the new strand. So this is the new strand, the green strand. Except DNA polymerase, look, he made a mistake right here. Is C and T supposed to go together? No, what's supposed to be there? G. So, and he did catch his mistake. He's down, he's down the line, working, right? And so before this gets methylated, other enzymes come along and they check the code. And they recognize and they check the code on this side to see if it matches with this side. And it doesn't at this point, right? It gets here and he goes, uh-oh, we have a problem, right? This isn't matching up. So these enzymes bind, these proteins bind, and they will literally cut out that section, right? So they bend over the DNA and they clip it and they cut it out. And then DNA polymerase has got to come in and remake that section. And then another enzyme has to paste the pieces back together. Because the DNA ligase has got to put the backbone pieces together. Because you're going to end up with a little gap right here. So that's mismatch repair. So what type of mutation is that we just fixed? Where C doesn't match with T. What do we call those C's, T's, G's? What are they? They're a part of the nucleotide, right? What part? So you have the phosphate, you have the sugar. What's the A's, the T's, and the G's? What's the chemical name for them? Nitrogenous bases. So for short, we say it's a base, right? So this mutation is a base, or you put the wrong base. What do we call that? A base substitution. So we fix base substitutions by mismatch repair, right? When they don't match, we're going to cut it out, that section, we're going to replace it and glue it back together. Before we methylate it, right, we check it. That's what mismatch repair checks and fixes, is those base substitutions. So, of course, I already told you, right? What causes these thymine dimers? UV light. Can we fix it? Sure thing. There's an enzyme known as photoligase that binds the DNA. Anyone know why it's called photoligase? What does photo mean? No. What does photosynthesis use? Light. So guess what we're going to use as energy to break this bond? Light. And a lot of times it's called direct repair or light repair because it directly fixes it. It's not going to cut anything out. It binds to that section. It uses the energy from light to break these double bonds, this covalent bonding between these two T's, so they go back to hydrogen bonding with the adenines across the way, and you get rid of this distortion in the backbone of the DNA. Those of you guys that were, oh, you stepped out or were late. If you were late, you need to email me today and let me know you were late. Otherwise, I won't fix you on the roll. I don't know what I did with my little book. All right, so that's pretty much the end of Chapter 7. Originally, when I had posted this, I accidentally highlighted transposons. Those are in the next chapter. So we've gone over these, but we've got transposons and knockout mutation that's going to be in Chapter 9. So remember, as we talk about each one of these types, we need to know how they form and their potential effects on a cell. So just as a little review, spot the mutation. So what mutation is this and what causes it? This is thymine dimers, and UV light causes it. And another reason why I picked this diagram is notice methyl groups. This is on mature DNA, right? And how would we fix this? Photoligase, right? That photoreactive, that enzyme will bind, it'll use light energy, it will break this double bond and restore the structure and the functionality of the DNA. What's this one? Diagramming. What type of mutation is this? So here are our choices, right? Base substitutions, silent mutation, missense, nonsense, frame shift, induced by chemical mutagens, radiation, by UV light or X-rays. What mutation is this?
Frame shift.
This is frame shift. Yep. By adding a base, it shifted the reading frame. Y'all see, the rest of y'all see that now? This is a frame shift. This is how it's supposed to be. This is the change, the mutation. What was done here? This isn't one of the lists. This is one of the exceptions to one of the things on the list. Where a frame shift doesn't happen, instead I added what? There's two correct answers here. What did I add? A whole what? A whole codon, right? In the RNA or DNA, and therefore added a whole, what are these? 3 letter representations. So this is what? What's the first strand? What code are we looking at here? Is this DNA or RNA? This is DNA, right? Because of the T's. This is what the messenger RNA would say, right? And this is a 3-letter abbreviation for what? We go from DNA to RNA to what? Proteins. So what are proteins made out of? Amino acids. This is that exception where you add a whole amino acid, right? And you don't get a frame shift. This is rare, but it can happen. All right, so this is what it's supposed to be, the wild type. Supposed to be tyrosine. There is a mutation that happens in this codon in the DNA. So therefore, as you can see, the codon in the messenger RNA has changed. But the amino acid's the same. So what mutation is that one? Silent, yep. For this one, we've got a mutation, right? The DNA has changed, the codon has changed, and the amino acid has changed. Which mutation is this? Missense. Again, a mutation, a change in the DNA, a change in the codon, and now it codes for a stop, where the ribosome is going to stop. This is the nonsense mutation. What could cause a double-strand break in DNA? I mentioned it. We were having a discussion about why you wouldn't want to go and do this all the time, because it does this to your DNA. Not UV light, another type of radiation, X-rays.
So DNA would be like for chemotherapy? Is that when the DNA gets affected by radiation?
Well, chemotherapy means a chemical therapy. So usually they're giving you some type of drug. And typically that type of thing affects the cell's ability to reproduce rapidly. Stops highly reproducing cells from reproducing rapidly. Radiation that they can do for treatment, right, of rapidly producing cells like cancerous cells. Again, yes, that's why it's effective and why you want to try and target it. Because all those cells they irradiate at that area and that level of radiation, they're literally chopping up the DNA. So those cells can't reproduce anymore. And so anything in that path also gets chopped up. Make sense? Yeah, that's why, you know, we're trying the best we can to develop therapies that are more what we call targeted, right? Where you're only targeting the cells that are bad and not everybody else who happens to be nearby. And that's the problem like chemotherapy, is that, again, it just pretty much for the most part targets cells that are rapidly dividing. So other cells are rapidly dividing that you need that get decreased in numbers because the chemotherapy stops them from reproducing. So like red blood cells, that's why they tend to become anemic. White blood cells too. And that leaves you predisposed to infection. So that's your defense. So the last thing that I want to talk about before letting you guys go today is in the modules. So again, we still got quite a bit of genetics left, unfortunately, and I'll post that lecture as soon as possible, hopefully by Thursday.
What you did was you updated that PowerPoint, so that's a PowerPoint edition.
Yes, that PowerPoint I just showed you guys, that's the genetics B that I posted under the objectives, right? So that,
All we have to do is add pages to the...
yeah, you could just add the pages that I added if you want. Yeah, you don't necessarily have to print the whole thing if you've already printed it. So the other thing that we have that you guys are supposed to mostly work on your own, and I realize that these instructions aren't very good, and so I'm going to try and write them better. But also try and explain it to you guys to help out. So there is 6 case studies that you need to do outside of class, and then you'll take a test on them. But one of the case studies, I don't give you the answers. The listeria one. So here's the questions that I've modified from the actual case study itself. This is the actual case study. And then this is another book that you could read to find answers and learn about stuff. And then this is an assignment that you will use, and you can use your notes to answer these questions. And then it will tell you the answers, right? So you will know the answers before you take the test. The rest of these, there's the case study itself, that other book.
Is this the one you're talking about that's all recorded?
Yeah. This is another document, I can't remember what. Here's a PowerPoint that goes over most of the answers. But I used to go over these in class. But we've added different content to this class, so I don't really have time during class. Instead of giving you guys a research project, I do the case studies. Students seem to really like them. So this is the PowerPoint, but under the instructions, I have recorded lectures that you can listen to.
And this is one attempt on these.
One attempt on the assignment for Listeria, yes. Once you open it, that's it, and do it, because it's going to tell you the answers. Same is true for the test that you'll take on all of them, which is on the computer. You can look at your notes. Heck, you can search the internet if you want to, right? It's just basically so I know you went over the material, okay? It's not going to be on the final exam. You can even do it after the final exam, but be working on it now, get it knocked out. I'm actually going to refer to the hepatitis one a lot when I talk about viruses. So, if anything, if you're going to start working on this, I would start by looking at the hepatitis one, because I will refer to it when I talk about viruses. One of the example viruses that I talk about. And after we finish genetics, that's what we're going on to is viruses.